Welcome to this ScarfWeb video tutorial on how to make use of the cell surface markers in CITEseq data to select and explore specific cell types. My name is Göran Karlsson, I'm the co-founder and head of partnerships for Nygen Analytics. When you have logged into ScarfWeb, you end up on the project page. Here, select the dataset that you want to analyze. Today we will analyze a publicly available dataset from the Greenleaf lab, where they have performed CITEseq on mononuclear cells from peripheral blood and bone marrow, as well as from stem cell-enriched populations. We will see if we can use the sequenced cell surface protein barcodes, referred to as ADTs, to fish out the rare stem cells from this dataset and reveal their gene expression program. This dataset has already been analyzed. For a tutorial on how to analyze your data using ScarfWeb, please see our dedicated video tutorial on this in our channel. To get to the Explore page, click View. You can now see a view map of the entire dataset, including cell clusters. If you go to Features, you can highlight cell surface ADTs. If you scroll down, you can copy all ADT names for use in another feature. For example, as an overview, it can be convenient to visualize all ADTs in a dot plot. Go to the dot plot feature. Paste your ADT list in the bulk editor, and you will get the expression of all ADTs in the different clusters. The dot plot is customizable. I will flip the axis, so I have the cell surface marker on the y-axis. I then move the stem cell markers next to each other, and I then change to a more fitting color, and also remove the size indicator for the number of cells that express the cell surface marker to only focus on expression levels. When I'm satisfied with my dot plot, I can just download it and directly use it for a presentation or a manuscript figure. Going back to the UMap, I will now select and highlight a few marker ADTs for mature hematopoietic populations. Remember that all graphs and figures in ScarfWeb are customizable, so change around colors and background until you have the figure you like. I will take a snapshot of a number of highlighted markers for use in my figures. Finally, I highlight the stem cell markers CD34 and CD90 to identify the stem cell containing cluster. To dissect the stem cell containing cluster, I use the lasso tool to select it and save it. Let's call it CD90 positive hematopoietic stem cells, or HSCs. I can now extract these cells into a new dataset containing only the stem cell containing cluster. While the extraction of these cells into a new dataset is ongoing, I will download my UMap screenshots. The extracted cells will end up as a new dataset in the project you're working in. Here you will need to reanalyze them. When reanalysis of the extracted dataset is done, click on View to get your data. You now have a new UMap containing your extracted cells reclustered. I will now use the known stem cell markers to isolate the stem cell population. First, I highlight CD34. If you go to Adjust, you can now select cells depending on their expression of CD34. I use the handle to select only the CD34 highest cells. Confirm and save your selection. If you go to Saved Populations, you can click on the eye and these cells will be selected again. I can then move on to CD38. I will go to Adjust, and knowing the stem cells express low levels of CD38, I will now refine the stem cell population by removing CD38 high cells. Note how you can see in the UMap which cells are selected as you change the parameters. Again, save your selection. The CD34 positive and CD38 negative population can be further purified for stem cells using CD90. So we do the same thing again, but now we select CD90 high cells. However, there is an obvious heterogeneity still in this population. So we group the cells by cell cycle phase and remove the cells from my CD90 positive selection that are actively dividing. So now I have a putative stem cell population that is CD34 positive, CD38 negative and CD90 positive and non-dividing. To be able to compare this population with something, I will also create a CD90 negative MPP reference sample. Now I will ask what genes that make up the molecular signature of my stem cells. So I click Comparisons and I select to compare the HSCs with the MPPs. Click Compare and a differential gene expression analysis of my populations will start. It will take ScarfWeb a few minutes to perform this task. When it's done, you can either visualize the results as a table or a volcano plot. In the volcano plot, click on the dots to see the gene names. As you can see here, our putative stem cell population contains all the expected stem cell markers like AVP, MMLT3, and HLF, while the MPPs have already started to express some lineage-specific markers like MPO. When you're happy with your volcano plot, just download it in the format you prefer and use it in a presentation. The downloaded data from ScarfWeb can be directly imported to your presentation or your manuscript figures. With ScarfWeb, you go from data generation to data figures in just a few hours. I hope this tutorial was helpful. Don't hesitate to reach out to me directly on goran.karlsson@nygen.io or visit our webpage nygen.io if you have any questions or feedback. Good luck with your single-cell genomic analysis.